Don't mind everything opening up right now. It's the addendum. Open up this lab manual. I don't know. I should probably download it so I can edit it right. I thought it started on 19. Why did it go to 13? Oh, it's a PDF. I won't be able to edit it anyway. All right. This is a lab that has us basically going back and forth between the lab manual and our addendum. So if you have two monitors, which I do, but I'm only recording on one screen, feels bad, then it shouldn't be too bad. Um, so yeah, it's really just going from one to the other. So it starts us off with an introduction. All this stuff you should have already read. Um, we know histology is the study of tissues. And basically it consists four major types of tissues in the body: epithelium, connective tissue, muscle, and nervous tissues. We're going to start off with epithelial, which I think is the most complicated compared to the others. Remember, for things to be epithelial, they have to have an apical surface, which is open to like the free space, and a basal surface where it's anchored into underlying connective tissue. Um, at the base of most epithelial tissues is a non-cellular layer called the basement membrane, uh, which helps to attach it to the underlying tissue. Individual cells of the tissue are joined very closely to one another so that there is little to no intercellular space. Nice and tight. Avascular, so because of they don't have blood supply, they rely on uh, nutrients to diffuse and stuff like that. Um, they may develop from all, I don't really get this one. Epithelial tissues may develop from all three primary germ layers of an embryo, although they usually develop from ectoderm and endoderm. I have no idea what that's talking about. Anyway, it's got main functions here: protecting underlying tissues. If I'm not mistaken, they can get replaced fairly easily. So, use and abuse, right? Controlling permeability, so they're part of that absorption of substances and secretion of substances. That'll be stuff like, if I'm not mistaken, like the intestines, right? It's going to be absorbing shit into the tissues, or it could be like secreting mucus to protect the tissue. And of course, transporting stuff over the surface using its cilia. Um, yep, we get into different cell arrangements. So there's the one cell layer, like these guys, one, 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 easy peasy. There's the stratified, which they're stacked. We know what that is. Stacked up nice and tight. And then there's the pseudo stratified, where they appear to be stacked, but they actually touch the bottom, and that would be letter D here. That columnar, if I'm saying that right. The shape of it, squamous, that's what we went over in class, where it kind of looks like a fried egg from the side. It's very flat. Maybe it has a little hump nucleus inside. There's little distance between the apical and basal surfaces. Again, that's all part of diffusion. Less cytoplasm to go through. Cuboidal is the cuboidal cube shape. Um, it's as tall as it is wide. Columnar obviously looks like a column, and transitional can kind of go back and forth between the two. between cuboidal and squamose, depending on the degree of stretching or distension. So if it's not under any stress, it'll look like a cuboidal, right? But if you stretch it out, like left and right, for example, it'll stretch and be like a squamous, but it can go back and forth without any like permanent distortion. I don't know how to recognize that. So here it says F is a transitional. I mean, looks like a cuboidal to me. So yeah, that is gonna be a hard one. But anyway, now that we have a basic understanding, quick review, we're gonna get into the actual lab part. And ideally, this is in person, right? And you're going through microscope slides, and you're like, oh, look at this thick kidney, and look at this, like an intestine slide or thin thin kidney slide. Because it's online, we have to basically flip to our screen, which has like a bunch of different slides for us to look through. So bear with me. So first, it wants us to look at a simple squamous epithelium. The slides are going to be kidney thick as well as small intestine. Let's see if we can find that. Oh, look at that. Easy peasy. Um, how do I not have this do this? Download it? I'll download it. I'll try to download it. Alright, well, that's figuring it out. Alright. The simple squamous epithelium. It can be in other places, but the slide it's showing us is the thick kidney and the small intestine. The simple squamous epithelial tissue is composed of a single layer of flat cells. Because they're flat, they're really hard to see, and depending how these slides are made, we're not gonna, we're just not gonna see them all, right? The function of it, like I said, is absorption of substances through diffusion or osmosis and filtration. It lines the body cavities and function in secretion. Other like parts of the body that's in alveoli in the lungs is a oxygen carbon dioxide transmitters, I think. Bowman's capsule in the kidney lining of blood vessels, heart, and body cavities. So it's taken from the outer portion, the cortex of the kidney. Look for the structure referred to as glomerulus capsules in the atlas. Note that cells are very thin and that the nucleus cannot be seen in each cell. So maybe we can make this even bigger. Hell yeah. So like this would be a squamous. We can see they're like super flat and they're kind of surrounding this this whole thing here. These flat little discs. So like these round ones wouldn't do it. These other like ones over here, I don't think they're gonna do it. It's only gonna be these super flat ones that we're gonna say are simple squamous epithelium. And then so like this is the same thing, but it just has images posted in it. Kidney slide image, kidney thick section slide. So there are differences. It doesn't say thick, but it doesn't say thin either. So I'm just gonna use this one really, um, because because then after you have the two slides, it goes to cuboidal, and this one has the thick. See, this is showing two slides. I don't know, man. I don't know. Anyway, then it goes to columnar, cuboidal. So yeah, fuck it. I'm just gonna use this one. So we're just basically going through this and trying to identify what we're supposed to be looking at. Small intestine slide is a cross section of the small intestine. If you look at the slide with your naked eye or 4 times with a microscope, it will look like a donut with a highly folded lining on the inside hole, or lumen, through which food would move through. The simple squamous epithelium will be found on the outer surface of the intestine, which would be facing the body cavity, not the lumen. Whoa. I really wish they could zoom this image out. Because in my mind, this is lumen because it's a giant space. But they're saying it's on the outside of the gut. So I would imagine, like, if this thing kept going down, like, there'd be a big hole here where food would be in the bottom. And this is just the outside of the gut. Yeah, it makes sense, right? Outer surface. But regardless, we know it's simple squamous epithelium because of the small pancake-like shape. So that takes care of the simple squamous. We're moving on to simple cuboidal. Cuboidal, obviously they're shaped like cubes, cube-shaped, yep. They will appear in microscopic views as squares with slightly rounded edges. Functions, the primary function are absorption and secretion of substances. That's weird, because this one is primary absorption of substances. So it also functions in secretion, but I guess this is absorption first, secretion second. This one is like half and half. Yeah. Yeah, I'm going to say half and half to try to make sense of that. Location in the body. It composes the kidney tubules. Many glands have this. Exocrine ducts and thyroid. Additional comments on microscope slides for simple cuboidal epithelium. Uses kidney thin section slide. The simple cuboidal cells will be found lining tubes within the kidney. Depending on where you look, you will see simple cuboidal epithelium in cross section with cells in a circular arrangement, or longitude section with cells arranged in long parallel arrays. So that's just how we're looking at them, I think, is what it's trying to tell me here. So those are columnar. So where are the simple cuboidal epithelium here? I mean, it's kind of cone shaped more than cubed, right? Lining tubes within the kidney. So these look like tubes, I guess. And they sure line it. And this is a bigger view. I really wish they put like times 100, this like times 1,000. Hmm. View as squares with slightly rounded edges. I don't know, man. I don't know. It's kind of rectangular. My thing is, how do we know that's not like a columnar that's going down, right? That looks like a columnar to me. It's longer than it is wide. I don't know. I don't know. Anyway, moving on to simple columnar epithelium. Maybe it'll make sense once we see these. The epithelial tissue is composed of a single layer of column shapes, which are viewed as rectangles, not squares, with slightly rounded edges. Everything has slightly rounded edges. Its primary function is protection and absorption and secretion. So a third protection, a third absorption, and a third secretion, I guess. Locations in the body composes the lining of the stomach, intestines, and excretory, excretory ducts of many glands. Using the same small intestine slide that you used to observe simple squamous epithelium, you will now look at the highly folded inner lining of the organ. Excellent. We saw this one in class. I should probably continue reading this real quick. Define simple. Yep. You may need to look around on the slide, find an area that has been cut in a true cross section so that it is accurately seen in a single layer. If it's cut like in an oblique angle, cells will appear to be layered when they are actually not. Once you find a good example of the simple columnar cells, examine the cells with the 40 times objective and note the apical surface of the membrane appears to be thicker. What you're observing is numerous folds of the cell membrane called microvilli, also referred as brush border. That's an interesting term I've never heard before. The microvilli increases surface area of the cell membrane to enhance absorption by the cells. Remember reading that it gives like 250 times more surface area? Scattered in between some of the columnars, you'll find goblet cells having a translucent appearance. They are unicellular glands that secrete mucus. The translucent appearance in the mucus that has accumulated before being released. Alright, so yeah, it's pointing these out to us, columnar. Easy peasy, right? Wonder if it's going to have like all of them side by side. That would be very helpful. But yeah, so these this is that the nucleus of the columnar? It's gotta be, right? And then like we can't see, but there should be a bunch of micro or mitochondria, and then there's like a basement layer that connects them all, and then there should be connective tissue, right? Don't know. Stratified, so there should be layered squamous, pancakes, eggs, bacon, something like that. The rule for classifying cell shape for stratified tissues is to look at the shape of the cells closest to the apical surface of the tissue. Thus, when stratified squamous epithelium, the shapes of the cells closer to the basal surface of the tissue are actually cuboidal in shape. However, moving up from the basal layers of the tissue, cells gradually become more flattened in appearance, so the cells in the layers toward the apical surface have a truly squamous shape. Sloughing. I don't even know what that is. What is sloughing? Shed or remove. It's sloughing. That's my new sloughing. That's my new word of the day. Let's go. So it's saying I can't zoom any more than this. It's 500%. Um, so it's saying like these are more cuboidal, even though we can't see like actual things. But as they get closer to the edge, they flatten out. Yeah, that makes sense, right? Anyway, its function is protection from abrasion. Okay. Compares the epidermis of skin, or composes the epidermis of skin, mouth, esophagus, other surfaces, cornea of the eye. Transitional, so these are the in-betweens, go back and forth. Many layers with varying sizes and shapes, round, cuboidal, flat, or irregular. The variability this feature is used to identify the shape, even though it always stratify. The term is never used in the formal name, since there is no simple version of the cell that it could be confused with. It is ultimately protection by allowing a high degree of stretching to accommodate changes in the size of the lumen. So yeah, any organ that stretches. So urinary system, lining of the uterus, urinary bladder, parts of the urethra. And then it's got a picture here for us. Hmm. It's very strange. But I think I kind of see what it's doing. So like these arrows are pointing this way, but ultimately these layers are going out toward the lumen, right? Like up and down. We can see that there are all kinds of different shapes and sizes. Normally, if it was like stratified pancake, egg, squamous, right? You'd have kind of rounder, and as they got closer to the edge, they would be super pancaked. But you got like some pancakes here, you got some round ones here. It's all over the place. We got pseudostratified, ciliated, columnar, epithelium. Holy cow, that's a mouthful. Looks like the slide is of the trachea, the thin section. It's technically simple because cells touch the basement membrane. However, cells have different heights, so some cells bend over others, and only some of the cells reach the apical surface. That is interesting. Because of the tissue, because of this, the tissue gives the appearance of being stratified when it's really not, hence pseudo-stratified. Its form function is protection. Because of the cilia present, the tissue also functions as transport of mucus over the cell surface. Because it has trapped debris, this transport function plays a role in protection. Okay, that's kind of silly, but I'll take it. Location in the body, portions of the respiratory tract, nasal cavities, larynx, trachea, bronchi, parts of male reproductive tract. Wow. And then our picture here, this slide has both similarities and differences compared to the simple columnar epithelial slide that you previously observed. Similarity is that you will notice translucent goblets interspersed between some of the ciliated columnar cells of the tissue. Major difference is that you will observe long, thin structures on the apical surface of the cells called cilia, while the simple columnar epithelium has microvilli. Okay, that's the difference. It is important to note that cilia are different from microvilli in both appearance and their function. Cilia are longer than microvilli, and their function is for transport. All right. So we got a goblet cell. We got pseudo stratified ciliated columnar epithelium. And those have those long cilia to move mucus and stuff around. Nice. Yay, we're on the connective tissue. Areolar tissue. This is the typical connective tissue found in association with it. Describes the pattern or weave of fibers in an open framework. Areolar is open framework. Easy. Areolar tissue fills up spaces between other tissues and provides cushioning for organs. It also provides a stable environment for cells involved in inflammation and fighting infection. It's found directly under all epithelial tissues, and it also makes up part of hypodermis, subcanthus tissue, and is found in the walls of blood vessels that penetrate other tissues of the body. All right. So all these strands are just collagen fiber. Well, no, because you have elastic fibers as well. How do you tell the two apart? I don't know how to tell the two apart. Darker is the only thing I can say. Is it's darker. Fibroblasts are the weird ball-looking things. Hmm. Yeah, it's thinner and darker, I think. So the collagen would be thin and dark, and it looks like the fibers are all of this darker stuff that's thicker. Mainly because if you look over on this slide, you'll see like there's all these kind of purplish ones, and they got these big red ones. I don't know. We'll have to ask her. What is the difference between collagen fibers and elastic fibers? Mainly on how they look. Adipose tissue is composed of adipocytes. These are found in areolar tissue. Each cell accumulates fat in a large droplet. The fat droplet enlarges so much that it presses into the cytoplasm and nucleus of the cell up against the cell membrane, forming a thin outer rim of cytoplasm in the cell. Due to the alcohols used in the staining of the tissue, all of the fat is actually removed from the droplet so that it appears extremely clear on the slide. Ooh, okay. So if we see clear, we just want to imagine a giant fat droplet. Adipose tissue is very vascular, so it has a lot of blood vessel flowings. It's weird, I thought fat didn't. Unlike other connective tissues, however, it does not have much intercellular space between cells, and only, or inter cell space inside the cells? I don't know. And only blood vessels and a few collagen fibers anchor the cells together. Its function: fats serve as the major energy source of our body. They also provide insulation, conserve heat, found in a liquid state above 37 degrees Celsius. They form a unique cushion for protection or padding for various organs. Yeah, here we go. Fat cells. McDonald's. Taco Bell. Some of the predominant locations for adipose tissues include the hypodermis, which is underneath the skin, located just below the integument skin, between the eyeball and the orbit of the skull, around the kidneys, and in the marrow within long bones. So these appear clear, but they're supposed to have a bunch of fat inside of them. And the nuclei has been pressed all the way against the membrane. Okay. I can point out a fat cell, let me tell you. Seen it. I've seen it with my own eyes. Dense regular connective tissue. It's going to show us the slide of a tendon. Dense regular tissue differs from areolar tissue in that present the only cells present are fibroblasts, and they are relatively few in number. In addition, the collagen fibers of the tissue are closely packed and are organized in a parallel arrangement, hence the name dense regular. There's very little ground substance present. This tissue is very rigid and does not allow much compression or stretching. Functions: dense regular connective tissue provides a strong and supportive anchor between specific body structures. Locations, it composes tendons that connects muscle to bone and ligaments which connects bone to bone. Those are the two major ones. So tendons are from muscle to bone and ligaments are going to be from bone to bone. So it's dense between these arrows, yeah. And I guess the regular is just like they're all going the same direction. Dense irregular connective tissue is the going to show us pigmented skin. Dense irregular tissue consists of collagen fibers woven together, oriented in multiple directions. The fibers are not as tightly packed as those found in dense regular connective tissues, so that more ground substance is visible in dense irregular connective tissues. So I guess we're looking for more dense ground substance. Is that cement, if I'm not mistaken? Dense, irregular connective tissue provides support and resists distortion in multiple directions. Probably because they're not all going the same direction, right? Easy to understand. Composes the reticular layer in the dermis of skin. Perichondrium around the cartilage. Periosteum around the bone. Capsules around visceral organs such as kidneys, spleen, lymph nodes, adrenal glands, the dura mater, I don't even know what that is, covering the brain. I think all bones have like a coating, a covering around them. Let's see. Most authors state that loose is simply loose. There's no regular or irregular. It is, however, either areola, adipose, or reticular. Some authors do describe loose connective tissue as loose irregular connective tissue. Okay. Just dropped the bomb on my brain. I thought we were looking at dense irregular connective tissue. This isn't dense at all, right? Unless it took this section and zoomed in, so I guess it is dense, but it's all over the place. It's not in neat little lines. It's just interwoven like crazy. Okay, okay. We're getting through. We're, I think, a quarter of the way through. Maybe we're further than that. Let's give ourselves some credit, okay? Elastic tissue. It's going to show us a slide of the aorta, artery, vein, nerve, elastic connective tissue. In elastic connective tissue, the elastic fibers are the predominant fiber type and appear as wavy arrays. The fibers are not quite as tightly packed or as regular arranged as those dense regular connective tissues like tendons and ligaments, I'm assuming. Um, and there's more ground substance present. The aorta slide shows portion of the artery wall, which is composed of elastic fibers intermixed with smooth muscle. You will see the wavy elastic fibers appearing in small patches arranged around the circumference. Okay. Composes the ligaments between vertebrae, vocal cords, walls of large arteries, trachea, bronchi, lens of the eye. So, okay, I got it. It's going to be wavy. I think it's going to be wavy if it's not under tension, right? But if you were to pull it, they would straighten out, and it'd be very difficult to tell them apart from the dense regular. But this is dense connective tissue, not regular, not irregular elastic tissue. Cool. All three side by side. Here we go. Man, they need to have these for every single one. What the heck? So we got wavy at the top. See the waves. We got the dense regular because they're all going in the same direction. Dense irregular is all over the place. Sweet. Supporting connective tissue cartilage. Oh, we're already on the cartilage. Let's go. So it's cartilage, bone, nerves. Yeah, something like that. Wow, there's a lot of bold stuff here. Supporting connective tissue cartilage. There are three different types of cartilage. Each is named for its respective fiber type. So we're looking for fiber type. But they all have several things in common. First, the matrix is firm in consistency. It provides support protection, but it still has some flexibility. The cartilage cells are called chondrocytes, like a hypochondriac, chondrocytes, are located in small cavities called lacunae. Oh, lacuna? Lacunae? I'm lost. I'm going to call it lacunae. Sounds better. The chondrocytes undergo mitosis, so that's cell division, right? As the cartilage grows, but as the matrix becomes more viscous over time, the dividing cells are unable to move apart. They become trapped close together, forming small aggregates or groups of chondrocytes in their individual lacunae. These aggregates are characteristics of all cartilages. So all cartilages are going to divide and split and grow, and eventually its matrix is going to become too viscous and they're not going to be able to move far, and they're just going to be boom, here we go. Cartilage is covered by a structure called the perichondrium, composed of two layers. The outer fibrous layer of the perichondrium is composed of dense, irregular connective tissue. The outside of it is, which provides support and protection to the cartilage. The inner cellular layer functions in growth and maintenance. Okay, that was weird. It says the outer layer of the perichondrium is composed of dense, irregular connective tissue. And that function of that structure provides support and protection to cartilage. But then it says the inner cellular layer of the perichondrium functions in growth and maintenance. This layer is the site where new cartilage cells are formed. In addition, since blood vessels do not penetrate this matrix, nutrients must diffuse through it from blood vessels located in this layer. So it doesn't tell us what the layer is composed of. Outer fibrous layer, but the inner cellular layer, it's composed of cells, maybe? Yeah, it's gotta be. Okay, so the outer fibrous layer is connective tissue, dense, irregular connective tissue, while the intercellular layer is probably simple squamous or something, that connects to that connective tissue. I think I'm starting to get it. I don't know why they couldn't just put something like that. It would help my brain. In addition, blood vessels do not penetrate the cartilage matrix, so they have to get their stuff through diffusion. Makes sense? No slides of cartilage? Oh, they're all the way down there. Okay. Hyaline cartilage. Trachea thick section. This is a section of the trachea. It is actually the same slide that you looked at for our pseudostratified ciliated columnar epithelium. Wow. You will just be examining a different portion of the slide. Hyaline means clear, and this refers to the matrix of the cartilage where there are no visible fibers seen within the ground substance. The different shades of color seen in the matrix are due to stain accumulating around the lacunae. In an intact tissue, the chondrocyte fills the lacunae. But during the slide preparation, shrinkage occurs. The Chondrocyte secretes ground substance and thin collagen fibers, which both will have similar consistency and thus cannot be distinguished from one another on the slide. At the outer edge of the cartilage, you will find the perichondrium. And the perichondrium has two layers, an outer fibrous and an inner cellular. All right. While examining this slide, also look at the pseudostratified ciliated columnar epithelium at the outer surface of the trachea, as well as the areolar tissue just below it. Holy cow, we're getting big words here. Hyaline cartilage provides rigid support with some flexibility. It also reduces friction between bony surfaces. Thank you. That would have been really nice to have like way up there so I could understand. Like its function, right, is to reduce friction. Probably like a kneecap and other things that rub. It's the most common type of cartilage found in the human body. Is found in the major passageways of respiratory systems and at the ends of very long bones where they form joints. It also covers the ends of bones, providing the articulating surface of the joint. Note, the articulating surface is the only site where there is no perichondrium. In the fetus, hyaline cartilage makes up the skeleton before it is replaced by bone. Okay, that's what is this? What is this trying to show us? Right, that's what we care about. Okay, I'm seeing epithelium on one side. Perichondrium. I'm going to do perichondrium first. That's way up here. It's composed of two layers, the outer fibrous layer and the inner cellular layer. So looking at the perichondrium, the fibrous I think is on the bottom side and the cellular is on the top. That doesn't make sense. It would be the other way, right? Because it has to diffuse from, well, unless that's the lumen. I don't know, man. I don't know. That's the matrix. So they're able to not diffuse, mitosis and split, right, and grow. But as this matrix gets more viscous, thicker, and they can't move very far, then they just kind of group up. Dun dun dun. All right, moving on. I don't even know. Elastic cartilage. You will note the characteristic cartilage patterns of small aggregates or pairs of chondrocytes in their individual lacunae. What is different is that now there are dense blue-black elastic fibers in the ground substance. The fibers are so dense that you need to go to the edge near the peridium to see the curly individual elastic fibers. Okay. Elastic cartilage is more flexible than hyaline cartilage. It provides rigid support with greater flexibility to structures that may be bent or distorted so that they return to their original shapes. Found in areas of great activity as the external ear. I don't even know how to say that word. Eustachian tube, epiglottis, and the tip of the nose. Lacunae with chondrocytes that just look empty. And then elastic fibers are just between. Like, what if they're red? Would there just be a dense thing, or is dense just bigger? I don't know. Let's Maybe I should have been going through the actual lab manual. Looks exactly the same. Like it gives. These are talking about glands. We didn't get the glands, did we? We didn't. Ooh. All glands are multicellular. Endocrine, exocrine, halocrine. Dude, I don't even know. It's like defining these words. Adipose, dense, irregular, elastic. Supporting connective, hyaline. Elastic cartilage. I mean, yeah, and that's the end. So we kind of have to let's I guess try to finish this. So we got elastic cartilage by the elastic things. Oh man, this did not post the slide very friendly. You will not see the parachondrium in the particular slide because of the way in which it has been prepared. However, you should be aware that the fact that fibrocartilage, when intact, is surrounded by perichondrium. What you do see is a vast amount of collagenous fibers to the extent that you need to look for the scattered few pairs of chondrocytes in their individual lacunae. Function. Thick bundles of collagenous fibers in the matrix aid in preventing compression or absorbing shock, as well as increasing its ability to anchor structures to surfaces. Yes, okay. It makes up the joints and the intervertebral discs, menisci, and pubic symphysis. All right, so fibrocartilage is wavy and it's all over the place, so you're just looking for the lumens, not lumens. What are they called? La Lacrumbe. La oh jeez. Lacunae, which have the pairs of chondrocytes inside, which are clear because of how it's prepared. All three side by side. Oh man, I can't even remember what these were called. These were the elastic fibers. These were the fibrous cartilage. Elastic cartilage, fibrous cartilage, because of the very few spread out lacunae, but it's dense. And this one looks like lacunae are kind of spread out still. And yep, drawing a blank, no idea what it is. Let's go back up. Hyaline cartilage is what that is. Hyaline cartilage. Why it's dark. Anyway, that takes us to integumentary. Yay, we did it.